I'm so happy to be here. A lot of people said that, which means we're all happy to be here. That's good. Also, I wanted to start with just a little bit on why I'm happy to be here. And that's because the North Sea laterality meetings really have helped me shape my PhD work, which I finished finally last year. I started my PhD in 2014 with two supervisors in a lab that where I was the only one studying laterality. So I was very happy to meet another PhD student from another department, Tess Baking, who was doing her PhD with Ton Groothuis. So I got a laterality buddy and I got to tag along for all the laterality fun during my years there. And the first thing that I got to go to was the 2014 laterality, North Sea laterality meeting in Durham. And that's where I got talking to Mark Brisbaard, who really had a lot of ideas on how I could approach my research question. And in the end, it led to this paper. And he told me, well, you should not just go and test some people on this one lateralized function. We're past that. The thing that you should do is get a very large group of people that differ in terms of handedness, preferably also differ in terms of hemispheric dominance for language, and then have a bunch of laterality indices from those people and try and look at patterns. And I think we've seen so far from this conference and from the papers that we read from each other that more and more people are doing exactly that, which I think is really great. So I guess that paper would not have been there if I had not had that conversation with him in Durham during the North Sea laterality meeting. And there's another paper that would not exist if I had not presented this as a poster at the 2016 meeting. This was a whole bunch of replication studies that I did because I thought, well, I'm going to test this large group of people. I need to really be sure that what I'm measuring is what I intend to measure. So I need to replicate previous studies and see if these tasks worked. And I thought, well, just put them all on a poster so that it's out in the world in some way, and then I'll forget about it because I got the tasks that I need for my actual research. And then when presenting that poster, Marcus Hausmann and Chris McManus, they both left now. I was going to thank them, but they left. Okay, so they went up to me in my poster and they really motivated me. They were like, no, you should just submit this as a paper, just all the replication studies together, which was a really big project in the end, but I'm really happy that I did it. So I would not have done that if they hadn't motivated me. I think they actually meant submitted to a laterality journal, but the co-authors were not that familiar with that journal, so I'll blame them for it not ending up there. For the context of the study that I'm presenting today, I'll go a little bit into this study that I did in a larger group of people where I, within the visual perceptual domain, I got laterality indices for these 7 in total types of stimuli or aspects of stimuli that you could process. And I found different patterns for that. And in the correlations between these different laterality indices for these studies, for these functions, I concluded that the functional segregation that we see for visual words in the left hemisphere and viewing faces in the right is related to the co-lateralization of lower level functions, namely local detail processing in the left and more global level in the right hemisphere. Or you could also view that even at a more sensory lower level, namely spatial frequency content. And I also found that there was actually no relation between the visual processes that I examined and the landmark task, so spatial attention bias. Although I measured it obviously in the visual domain, it did not relate in any way to the other indices. So I think it's important to realize when we're doing studies like these, and these are our findings, we're really focused on which hemisphere does what. And sometimes we tend to forget that it's not that clear cut. And actually, it does not mean that the left hemisphere, for example, is not able at all to deal with face information, which would also not make much sense. For example, imagine that a potentially dangerous man is approaching from the right. Would that mean that it ending up in your left hemisphere would mean that you're in bigger trouble than when he were to come from the other side? So I think when you consider this, the next question is, okay, so, but there's clearly lateralization, so hemispheric specialization, preferred and non-preferred hemispheres to deal with some kinds of information. So when does that happen? And this question of when, you can interpret that in different ways. You can ask under which conditions does that happen, or at what point during a processing cascade. And just to give you some examples for face processing, there's a whole lot more, but it was shown that as task difficulty increases, the preferred hemisphere is recruited more. If people are sleep deprived, actually the non-preferred hemisphere tends to help, or that's how you could maybe interpret it. And when people are stressed, it's the idea is that just a wider network is activated, among which networks in the non-preferred hemisphere. And with regards to the question of what point during a processing cascade, actually, there's a word in the title of my presentation. It's unfolding, which we saw in Martina's presentation earlier this morning. And she also already mentioned for faces that around 170 milliseconds after the presentation of the face, already we see a clear dominance for the right hemisphere. And also the right hemisphere seems to stay interested in the face information much longer than the left hemisphere does. So what I wanted to do is to examine how the processing of different types of stimuli, words, faces, and spatial frequency content, how that relates to each other when we present it as task irrelevant stimuli. So a lot of these studies that we do are just people doing tasks. So they're engaging with stimuli, they're being asked questions about stimuli, being asked to do stuff with stimuli that we usually would probably not do, like categorize this face or recognize something about it. And it's not that a natural way to do it. So my experiment still is nothing like stuff that Sebastian and Gina are doing. It's just a lab EEG experiment. But I did choose a paradigm in which I could present the stimuli without people having to do anything with those. They were actually doing a target detection task that had nothing to do with the lateralized presented information that was that I presented. And I did that in an oddball paradigm that usually produces a mismatch negativity in the EEG signal. I really like how the chicks of Leslie Rogers keep coming up, so I have some chicks here, but just to illustrate the way that this paradigm works, it's when people get presented with a series of the same stimuli or type of stimuli over and over, and every so often, not too often, once in a while, they get an oddball, a deviant stimulus. And at that point, we see a negative deflection in their EEG signal. So that's the paradigm that I used for this study. And I'm only going to talk about the face information because I haven't had time to look at the other stuff yet. So I tested 32 participants actually, but 3 were excluded from the analysis. They had too many artifacts in their EEG. They were all right-handed, measured with the Flanders questionnaire for handedness, and they were students, so mean age 22 years. I used the oddball paradigm, and they did a target detection task. And just to show you the numbers of trials that went into the EEG analysis. So we had left visual field deviance and right visual field deviance, both around 120 trials, and then a whole bunch more for the standard face trials, because otherwise the deviance would not be deviance. And then there were 4 time times of interest that I looked at, one corresponding roughly to like the P1, one to the N170, and then two later ones that people usually look at when they use the visual mismatch negativity, or the like in this oddball paradigm. So the stimuli that I used were faces that I constructed to be symmetrical around the midpoint. They're just mirrored halves of two models from the Karolinska face database, I think this is. Yes. I had ones where they had neutral expressions, and one for happy and one for angry. I'm not going to zoom in on the emotional expression aspect of it in this study, or in this presentation at least, just using these as standards and deviants, because a change in information in the face needs to happen. And then these were the deviants. So either the left or the right visual field information would change with respect to the standard. Yeah. So this is what trials would look like. People would just be presented within a block. I should mention that. So each of the 4 standards would be a standard during one of the 4 blocks of the experiment. Right. So this would be a bit from one block where people have a standard, the neutral face of one of these models, and the target, I think maybe you can see, it's a circle with a cross inside. So whenever they spotted that target, they had to press a button. This is very convenient because it also forces people to centrally fixate the screen. And then there's an example of a deviant in the left visual field. So that's what participants were doing. Now for the results. This is the response as measured over posterior, so occipital, parietal electrodes in response to seeing the standard stimuli. Nothing too interesting about this in itself. But then if you look at what happens when a deviant appears in the right visual field, you can see that around 300 milliseconds, you can see both over the right and over the left hemisphere electrodes, you can see this negative deflection, meaning that the deviant has been processed or recognized as being deviant from the standard. And now if we look at the left visual field deviant, so everybody knows this, but the left visual field presenting, projecting to the right hemisphere, which in these right-handed participants, most probably is the dominant hemisphere for face processing. So what you see here, which is the pointer, because people seem to turn it off. Ah, I have it here. For the left hemisphere electrodes, you see that there's absolutely no difference with the right visual field deviant. But when measured over the right hemisphere, actually, you see that this left visual field deviant is prominent in the EEG signal very early on, actually, and it's more strongly a more strong negative deflection as well. This is actually more clearly seen from the difference waves when we subtract the deviants, the standards from the deviants. There's this one odd thing happening here that I'll get to now. If we look at the mean amplitudes for these times of interest, then as you can see, in the very earliest processing stage, as measured over both the left and the right hemisphere, the left visual field deviant is detected, the right visual field deviant not. And the hemispheres do not differ, but they do differ from the right visual field deviant. Then in the next, the second and the third stages, actually what is left is only the left visual field deviant, and only measured over the right hemisphere. So the left hemisphere at this point is no longer interested in doing anything with this left visual field deviant. But in the latest or the last processing stage that I took into account, basically because everything that's happening afterwards is just more of the same, you see that actually also the right visual field deviant now is processed. it can be measured both over left and right hemisphere electrodes, but it's still the left visual field deviant as measured over the right hemisphere that really pops out. Yeah, and here you can see that it's actually only, like in the previous ones, only this left visual field deviant over the right hemisphere that differs from the other conditions. To summarize, so changes in unattended phases elicit negative deflections. The paradigm works. In the earliest stage, the left visual field deviant is noted by both the left and the right hemisphere. The stages that follow, only the right hemisphere seems to still deal with that information. And then in the final stage, the left hemisphere is back on again. And now also only at this point, the right visual field deviant seems to do something. And I was thinking about why does this happen so early? Why is the left hemisphere involved? Why is it only the left visual field difference and not the right one? Because in the natural world, you wouldn't expect that. And I think that maybe this is something you see because, of course, in this experiment, they've been looking at faces over and over. So they get presented with faces all the time. So this is, I put your word in here as well, it's like prior, they seem to be primed. They know they have to deal with faces, even though they're not doing a task with the faces, they're looking at the face constantly. So this may prime them to be especially attentive, I guess, to the left visual field, although note that the left hemisphere is not sort of switched off here. It does note a left visual field difference. Next, of course, I want to see how this relates to the visual word perception that I incorporated in this experiment. And now going back to the relation between different lateralization of different types of information, I heard it mentioned before yesterday, and I thought it's interesting to note this, that there's this idea that when people learn to read at that point, the left hemisphere visual word form area gets specialized for that. And while the processing of faces was first bilateral, it now only remains in the right hemisphere just because that one is not dealing with the word form information, which was, it's probably around for a long time, but Marlene Beerman wrote a nice paper about this in 2015. But there's also a nice paper from earlier this year that shows that this support, the support for that theory is actually not that strong. And there's several reasons for that. One of them is that it seems to be based actually on only one study, a very nice study though by Stanislas de Hane in 2010, where he compared people who never learned to read with people who read. But the group of people who never learn to read was actually quite small, 10 people. And also this right shift, or sort of right hemisphere remainder for whatever is left, was not only the case for faces, but also for other stimuli. It was not specifically in the fusiform phase area, but more around. And the shift was for other stimuli as well, like houses. So nobody here speaks about the contrast between faces or words and houses. It's all words and faces. So it doesn't seem to really fit in. And I think one of the strongest pieces of support against that theory is actually that we see right hemisphere lateralization for faces in other species than humans, for example, chimpanzees, sheep, and rhesus monkeys and dogs. And obviously these animals never learn to read. And so why would they then have right hemisphere specialization for face processing? So that's the end of my presentation. These are the faces of my PhD supervisors who helped me with this work. And I'm on ResearchGate and Twitter and Open Science Framework, and I have an email address. So contact me or look up my data or whatever if you want to. And I'm very happy to take any questions or hear your suggestions.
Thank you very much.
Hi, thank you for that talk. I was just wondering, because you show the average, the group effect, right? So do you also see these effects on the individual level? So for each participant?
Right. Yeah. So because EEG data, of course, is a lot of data, you could, in principle, look at individual level. That's an interesting suggestion, especially when I get to relating words and faces and stuff. But I haven't had a look at that yet.
Because that is what Behrman did, right? She looked at EEG on the individual level.
Yeah, she does. She did do some EEG studies as well. Right. Yeah. Thanks for that suggestion. It's a good idea.
Thank you. That was really interesting. Bianca here. I was just curious with your oddball paradigm. Were there any trials where the oddball appeared at the same time as the deviant face?
Oh yeah, so it's a bit of a mix of terminology, but the oddball is actually the deviant face, but
Okay.
I think you mean the target that they
target.
had to respond to.
Yes.
No, the target would only appear during like standard trials.
Okay.
Yeah, yeah. So
Okay.
there's no conflicting information in the EEG signal in those trials.
Thank you.
And I compared the deviant trials only to those standard trials during which there was no target present. Yeah, so all of the tasks taken out.
Thank you very much.